So many partners are involved in work package 9.2, which is the work package on behavioral and physiological monitoring. Partners working across the entire animal kingdom in VetBioNet, and we've been very much involved with these partners, working on a variety of studies, of which the people shown on this slide will be presenting the results of the past year to you. And my task is to provide an overview and to do the timekeeping for all the speakers in this session. We have 8 speakers, 40 minutes, so 5 minutes per speaker, and a little bit less because we also want time for discussion. So the goal of work package 9.2 is to develop better tools for physiological and behavioral monitoring. And that involves the monitoring of a number of readouts. Body temperature is the #1, followed by general activity, movement, behavior, specifically eating and drinking behavior, and respiration. And we tackle them from top to bottom in the list. In the last year, at the annual meeting, we discussed the key requirements for the technology that we have to develop. For instance, when we talk about temperature measurement, we want a telemetric device that is reliable, that sends data in real time, and it does not migrate through the body. And when we attach something to the outside of the animal, we want a tag or a piece of electronics to be robust, tolerant to the natural behavior of the animal, small and lightweight, to accommodate the smallest animals being studied, for which that tag is relevant. And everything that we bring into the animal room should be tolerant to decontamination, to remove any microbes. Another objective of work package 9.2 is to come to a more objective definition and scoring in practice of the humane endpoint for animal studies. And that requires that behavioral and physiological data are collected in real time, and that the software collecting those data analyzes the data and generates automatic alerts to signal to the researcher that a humane endpoint has been reached. And third, we want to reduce the entries and exits of human beings in and out of BSL-3 facilities, and that requires that equipment must allow access to the systems from remotely from outside the hygienic barrier, and again that data analysis is carried out in real time, not requiring the supervisor operator to enter the animal room to read a machine. So those were the objectives agreed on in previous meetings, and in the last year at the annual meeting we presented a number of scenarios in which VetBioNet partners could access the technology that we are going to develop for behavioral and physiological monitoring. We described 2 scenarios. Either researchers can use these tools in a VetBioNet pilot study. That means they will receive them free of charge, and we will take care of installation. They will not become owner, but only use it for the duration of a study. That they will decontaminate, and we will do that out of a pool of equipment. Alternatively, the researcher can say, well, I want to have it now, I want to buy it, I want to own it, I want to have a perpetual license of the software, and I don't want to send anything back. And when we gave everybody the option to choose between one of the 2 scenarios, there was a unanimous vote for, you can guess, the first scenario, because everybody would like to try out for free with the budget from the VetBioNet project. So that has been the model that we've been following so far during the pilots that have been carried out and that are being planned. Then what did we do since last year? Since the previous annual meeting was in May 2019, and so from June onwards until today, we've continued our discussion with the VetBioNet partners about what needs to be measured, how it needs to be measured, how the data should be analyzed. And we've continued the development of new and improved tools in our R&D department. For the behavioral monitoring, we can do it all in-house because that's our expertise. For the physiological monitoring, we depend on external partners. We've had some internal developments, which were presented last year, but that specific line was discontinued, so we made a strategic choice to work with third-party technology for the physiological sensing. Now, in order to come to the right choice of partners, we are bringing in the collective expertise and intelligence, and also say the reasoning of all the partners in the consortium, all the partners involved in work package 9.2. And we're currently engaged in a survey among manufacturers of technology that Leslie, our assistant project manager, will report out about later in this session. The actual pilot studies in which these new and improved equipment is being tested and validated for infectious disease studies were severely disrupted by the COVID pandemic. And we'll show you an update on that later. But what we have greatly benefited from in the past year is the establishment and the routine meetings between 6 targeted discussion groups that we put together around animal species, in which we discuss technical developments, the tools prototypes, we show software and hardware demos, we present recent results, lessons learned, and we plan new pilot studies. And what you're going to see in the remainder of the 40 minutes are the outcome of these meetings, of the meetings that have been completed so far, presenting on the work that has been done in the past year. These groups are grouped around the animal species, which are logically grouped together because they have common requirements related to their morphology or behavior. So all ungulates have sort of similar requirements when it comes to tracking. They all have a neck in which you can attach something. The pigs are a group apart. Then we have the birds, the rodents, the carnivores, and the fish. The most heavily populated group is the pig group. However, all groups have sizable number of partners, and altogether we are working with more than 60 individuals from across the consortium in our daily interactions. So it has been very active, very energetic. So that's my introduction, and now I would like to introduce the first speaker in this row, David, who will be followed by Simone. They've been working together on ultra-wideband tracking, 3D accelerometer sensing, and automatic behavior recognition in cattle. So David, you're next. I'll handle the slides, and you only have to serve a speaker. Okay?
Okay, thank you, Lucas.
There you go. You just
Okay,
tell me when to move to the next slide.
Okay, will do. That's fine. So we've been having discussions with Noldus to trial 2 of their systems. One of them being TrackLab, ultra-wideband motion tracking of animals, and Viso, which is kind of a video capture system which can be used for later machine learning or downstream processing to look at animal behaviours. So background, we have high containment facilities here at Pirbright, SAPO4 and BSL3 ag, and we're licensed to work with foot and mouth disease. Within the building, we've got 14 animal rooms, so about 20 meters squared floor space, which can hold up to 5 calves for about 6 months old. Recently, we've installed 360 cameras in all of the rooms, and that's kind of allowed us to do some veterinary and behavioral analysis, and this kind of project is extending that. And to get, so one thing we had to kind of think about is bringing equipment into a biosecure envelope. So we wanted to bring as little equipment inside the building as possible. And some stuff we had to create to make waterproof, to allow it to be disinfected and fumigated following the trial. Okay, next slide, Lucas. Okay, so a very quick overview of the pilot study that we did. We had 5 cattle on a foot and mouth vaccination and challenge study. We kind of want, from the Pirbright Institute side of things, we wanted to look at the behaviours during the settling in. So we get the animals in, we'll hold them for 5 days to 7. The post-vaccination phase, so after they've had their first or second vaccination, and then also during the virus challenge phase. So we took some video footage before and after the virus challenge of foot and mouth. Okay, next slide, Noldus. Thank you. Okay, so the TrackLab equipment required some additional setup. So the way it works is you have anchors which are on the walls, which triangulate to a motion ultra-wideband transmitter, which is attached to the cow's neck via a collar. And what it was important to note is that we I think minimum you needed 4, but we had a blind spot in our room where the animals couldn't access. This could also apply if you have extra pens or bedding areas in the room. So we had to place 5 of the anchors around the room to allow a full triangulation of the animals. And then the Viso footage just took a live stream of the entire study period from the 360 camera, which we had already in the room. So that's a very brief overview of the study. So I'll hand over now to Simone to look at some of the results.
Yeah, before Simone starts, maybe, so this is the view of the overhead camera. And here you see the 5 neck collars just hanging on the rack, the food rack. These are the sensors, the tags that communicate with these anchors. And this is one anchor. These are the anchors attached to the wall of the room. Okay, I'll now click on to Simone, the AI researcher in our company.
Yes,
Simone.
Yeah, so we used the data from this pilot with the aim to automatically detect behaviors from this remote sensing. And there was a lot of data collected, and what we did was use a subset of this data and had human annotators make annotations for approximately 1 day for 2 of the calves, and combine these annotations with the signals from the neck collars, which contain the accelerometer, gyroscope, and location data at approximately 20 Hertz sample rate. And we pre-processed this, and it was particularly important to account for the fact that the data was recorded using a sleep mode. So whenever the animals were not moving, there was no data sent. So we had to account for that and segment the data into separate windows, split these into training, validation, and test sets. And we also applied data augmentation to the training data and tried to simulate for the noise of the sensors and try to make the model more robust. And on the right, you can sort of see the workflow where you go from raw data through the data segmentation and pre-processing steps. And then this goes into our neural network. Next slide, please. So for our neural network, we found that a relatively simple LSTM, so recurrent model, outperformed more complex models, such as a convolutional model. And on the right, you can see the accuracies that we reached on our test sets. So we reached relatively high accuracies for most of the behaviors. So you can see, for example, the ruminating and feeding behaviors had high accuracies. It was more difficult to correctly classify the drinking behavior, and this was probably due to the fact that there was not that many examples in our training set. And we also had some issues with walking. And again, this may be the number of events, but also the fact that it was a relatively small space that we're looking at. So the animals did not have like long walking events. And also because we're measuring using a neck collar. So whenever the animal's eating, then there's also a lot of movement, which may distort the signal a little bit. And we can zoom in on this locomotion behavior. If you could go to the next slide, please. Then here you can see the behaviors over time. And at the top, you can see the annotations made by the human observer using the VISO video. And at the bottom, you can see the predictions from the machine learning model. And here you can see that, so the walking behavior is not classified as accurately as we would like, but still this can serve as a sort of indication of movement, and yeah, it still provides insights into the behavior. So it's a step in a good direction. And what we will try to do next is look into further pre-processing, so how we can get more out of our data, and also see if we can apply additional data augmentation and sort of account for the fact that some behaviors occur less frequent than others. I think these are all the slides. Oh, no. As to conclude, to wrap up, we use this deep learning approach for cow behavior classification, and this leads to accurate results for most of the behaviors, and we're looking into improvements for the behaviors where we're not where the accuracies are a bit lower, and we are already improving our earlier results by having more sensors and also having additional pre-processing steps to account for the noise. Well, and the next steps I already went over, so that's about it on my part.
Okay, thank you very much, Simone. This is a very quick overview by David and Simone of how artificial intelligence, in particular deep learning, can help us to automatically recognize the behavior of cattle from a neck-worn sensor. Now we will make a jump to chickens, and Samantha Watson from the AFA will give a report on the pilot study carried out with lead-based identification of chickens in her institute. Samantha, the floor is yours. We really need to stick within the 5 minutes per speaker, so you may not want to discuss all the details on this slide. We don't hear you yet, Samantha. Yeah, I see you waving, but I don't hear you. We don't hear. Something wrong with your sound. Does anybody else hear Sam?
No.
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 No. So Sam, nobody hears you. Otherwise, we could continue with another story, and then when you fix your microphone, we go back to your slides. Okay? All right. So we stop. I'll move on to Maeva. Maeva, are you there?
I am, yes. Can you hear me?
Yes, I can hear you.
Great.
So Maeva usually takes a different approach on chicken tracking, and tell us what you have done. Here you are.
Yes, thank you. So yeah, I'm doing a PhD on laying-hen welfare, and I want to understand the mechanisms behind it. Next slide, please. So I'm focusing on the genotype and the incubation environment and their influence on behavior, neurobiology, and physiology. And within my PhD, I have a collaboration with Noldus and behavior part where we want to monitor movement. Next slide. Next slide, please. Yeah, good. So yeah, I want to monitor movement in chickens because so the genetic lines I use are
Now your sound is dropping as well.
Oh, can you hear me now?
Yes?
Yeah, okay. I'll just put on my camera then because I think there's a connection issue.
Yeah, okay.
So yeah, I'm looking at I think the movement monitoring can help me finding out about different social behaviors among the genetic lines and also find out about the additional impact the incubation conditions may have. And finally, I'd like to be able to monitor that from day one, ideally, until 5 months old, which is how long I keep the chickens, to know the evolution of their movements over time. Next slide. So first we started with pilots with flash track technology. I think Samantha was going to talk about it a little bit. But basically it gives the ID and the direction of the chicken, and it works with the camera and then the software EthoVision. Next slide. There should be a video, I guess. Yes, so it's in the form of a backpack, which you can see there. And the backpack is accepted by the chickens, and the tracking seems to work. But this backpack is of course way too big for a 1-day-old chick. So we wanted to try to find a solution which we could use on younger chicks. If you can.
Yeah, maybe you should explain that this backpack, this code generates a unique flashing code picked up by the video camera. And that should allow us to identify the chickens in a video tracking situation. Are you still there, Maeva? Quite a challenge with the technology. Let's move on to the next slide then.
I think we have lost her.
Yeah, we have lost Maeva, but while she is reconnecting, I can continue to talk, and then we'll see her arriving soon, hopefully. So the flash track technology was our first attempt at developing a identification method that allows us to track multiple chickens in a group using video tracking. Although the chickens all look similar, we need an identification method to tell them apart. And this electronic identification led to these backpacks that Maeva just showed, which were fairly large. So we were looking for something smaller and simpler and lighter. And then we came across the technique of 2D barcodes. And here you see a bird with a 2D barcode on its back, on a little marker. And these markers also can be translated into ID codes using computer vision technology. And that's the approach that we are following now with our next round of pilots. Maeva, are you back? And Sasha, are you still there?
I'm still there. I think Maeva has disconnected and she's probably trying to reconnect.
Right. I just want to make sure that at least some people are still there because I don't see anybody. But we will just continue, although if we run out of time. So these 2D barcodes come in different types. And one type that we all know is the QR code. The QR code that you find on many packages of products that you scan in the supermarket. There's also a micro QR code, whereby a number of, say, a pattern of black and white little squares represents a code that can be read by a video camera. But this is still fairly large. And there's also a smaller version of it. It's called Aruco. And these are called Aruco markers. And Aruco is very simple 4x4 bit pattern that still gives you endless possibilities for our type of studies. And that's what we are currently experimenting with. So here we will try to make these markers as light as possible so that this will be not much more than a piece of cardboard lying on the chick's back and attached to its neck or its feathers in some way. And then the test that we are now doing is, can we recognize these codes with the computer vision system, and indeed individually recognize it independent of the movement and the orientation of the animal. So one last check if Maeva is back. I don't hear her yet. Okay, then I'll. So what you're seeing here is not a test in an animal facility. It's a test at a researcher's home who set up his Christmas tree. But you see these little markers here. These are the Aruco markers, and the blue numbers are the ID codes recognized by the software. So our software has recognized these codes, and even if the codes are far away, well, at some distance he fails, but when you get close enough, we can recognize the code independent of the rotation, the zoom factor, and the angle of view. And that makes it opens up the way for a really robust tracking and identification system using these type of markers. So in the next weeks, we hope to turn this into actual physical markers that we can attach to birds, and then we will carry out in vivo studies with birds at Utrecht University, and next we go back to AFA in England. And so that's the art of Maeva's work. And now let's see if Sam can be heard by everybody.
Can you hear me now?
Yes, we can.
Yes.
So brilliant, brilliant. We changed over to a phone, so I can't see slides.
I go back to your slides, and then you give us the AFA story, okay?
Yep, that's fine.
Okay, there you go.
Okay. APHA carries out an awful lot of research on avian influenza in poultry. So predominantly we house chickens, and we do quite a lot of work on ducks, turkeys and game birds. APHA is also an OIE ref lab for highly pathogenic avian influenza, which involves characterization of avian influenza virus isolates. One of the tests that we run is called the intravenous pathogenicity index test, which is an OIE ref lab test to determine the pathogenicity of an avian influenza isolate, usually under bird flu outbreak conditions, to determine the pathogenicity of that strain. So the procedure involves you take 10 chickens and you inoculate them with 0.1 ml of 10 to the 6 of your virus isolates intravenous, and you monitor them for 10 days, scoring them daily, giving them a number from 0 to 3 on normal, sick, paralyzed or dead. The sum of that score multiplied by the number of chickens divided by 100 will then determine the pathogenicity of the avian influenza isolates, and a score of greater than 1.2 is considered high path. We also do a lot of other studies at APHA involving high path influenzas, such as transmission studies, transmission between species, so duck, chicken, that sort of thing. Next slide, please.
There you go.
Thank you. So, as always, when using animals in research, we want to look at ways to refine our systems and improve welfare. So one of our aims is to improve the detection of humane endpoints in high-path avian influenza studies, and we're interested in using motion tracking as a potential way forward. So poultry generally don't show clinical signs unless they are severely sick. So quite often, despite checking them up to 3 times a day, including a night check, animals may be found dead in the morning. Interactions with humans tends to mask any clinical signs. So we have, you can monitor them through an observation window where they can't see you and they look very sick. As soon as you walk in there to look at them, they will get up and pretend they're absolutely fine. So the requirement of our tracking system really was to have a system that would recognize a sick bird, primarily one that was moving less than the others, and then alert the animal care staff so that appropriate action could be taken. In the left-hand side picture, we have two kind of housing systems. On the left side there, those are our standard Perspex chicken housing that we use to house poultry. You can see they have roofs on the top to stop the birds flying out, but it's quite good visibility through the top. The other one is our game bird pens, which is mesh so that they don't hurt themselves, but again, you have good visibility from both the sides and the top. Okay, next slide, please. So what are the challenges facing the system we were trialing? Well, the system must be able to recognize individual animals so that we know which one it is that was sick. Because we work with high-path avian influenza, CL3 material, the system must be compatible with our decontamination procedures, which is primarily fumigation with formaldehyde. We needed it to be lightweight so as not to impact on the bird's natural behavior. And our facilities currently do not have network or Ethernet points. So we had to be careful that any alterations into the walls, drilling holes, etc., we would have to think very hard about sealability issues and affecting decontamination and cleaning afterwards. Okay, next slide. So we did a proof of concept trial using jacket transponders in chickens. So we took 10 healthy chickens to mock up our intravenous pathogenicity index test trial. We put them into a non-infectious environment, which was identical to our containment facility. So we housed them in the Perspex pens and we monitored them over a 24-hour period. The transponders were integrated into jackets that were worn by the chickens. A camera was placed over the pen to record the movement of the chickens, and this worked really well. And then a computer was placed in the animal room, which saved all the data. And next slide, please. So this slide just shows the chickens wearing their little jackets, and on the right is a setup of our animal room. So you can see the camera over the top, which was put on the ceiling without drilling any holes, so that was good, and the computer just sat to the side of it. I think I've got the final slide. So this was a very much early stage pilot, and what we discovered was that the jackets that we used were too heavy for the chickens to move freely in. So we put the jackets on, and they just hunched down and sat there for quite a considerable period of time. Again, because it was a pilot, the open electronics in the transponder made it unsuitable for fumigation. And at this stage, there was no ability to pick out individual chickens from the group. Also, having a computer located in the room is not ideal, but we didn't have a choice because we had no Ethernet. So that was our pilot study.
Okay, thank you very much, Sam. So you should all have seen these talks in a different order. First Sam's talk, and then my Eva followed from that, because we hope to go back to AFA with the Aruco codes, which should be simpler, lighter, and less than for the chickens. But I hope you understand that. So we will now continue with the next talk. And I was just notified by Sasha that we are running over time. So I asked the remaining speakers really to stay within the 5 minutes. And if you can do it in 4 minutes, that would even be greater. So Anna is next, talking about group-based video tracking in sea bass.
Okay, good evening, everybody. Can you hear me?
Yes.
Okay, fine. I will be really, really fast. This is just to show you how we work in our experimental aquarium. So we have 12 fiberglass tanks, 300 liter each. Each tank is an individual unit, so it's equipped with a mechanical filter, biological filter, heater or refrigerator according to the temperature we need, and obviously air or oxygen. Generally, we work with very large number of fish. The minimum is 30 fish per group, up to 130 fish according to the weight and the size. Of course, the smaller the fish, the higher the number. In general, we have a control group and then one or more treatment groups in other tanks. And we aim to compare the behavior and the disease development amongst group. The most used pathogen is the viral nervous necrosis, which is a virus. And we work very frequently with this viral disease. With sea bass, but also we infect fish with the bacterial pathogen like Vibrio and Tenacibaculum maritimum. So now we have a video. So please, Lucas, can start the video.
Now, yeah, will you or Fabrizio give comments?
Yeah, okay. So thank you, Fabrizio, for commenting this.
So this video was made by Fabrizio Rico, a behavior research consultant in our company, who's also joined the session. So maybe, yeah, Fabrizio, you can tell the story while the video does its work.
Yeah, I hope the video plays smoothly.
Yes,
the method is based on the change in the pixel intensity in the image. We define an area of interest, and then we try to set the software in such a way that only the movement of the fish is going to be detected, and not other object moving or particles of food or feces. So everything that has to do with the movement of the animal should be quantified in order to give a good estimate of the activity as a group. So we are not going to track individuals, we are going to track the shoal as a unity. And here the video shows how we can rather quickly set the software settings, and we take a look at the results online, and we do our best to remove any noise in the image that could cause an overestimate of activity. So the software will calculate all these purple pixels and give us statistics like the average the average amount of activity per time unit or per sample. And this video was shot with a smartphone, but it's of course possible to have a fixed camera and to record higher resolution. and to improve the lighting as well, as like we are going to say later.
Yeah.
Let's see, are we done with the video, Fabrizio, or should I move to the next slide?
Yeah, I think we can go to the next slide.
Because it seems as if the video stopped halfway.
It at some stage it stopped. So can you, maybe you can go. Yeah, now it's going.
Sure.
Yeah. Here we can see the amount of activity plotted against time. So here you can see, for instance, for long recording, whether activity is reduced or increased. Of course, it's activities depending on how many fish you have in this tank. So you have to correct the data some way. And yeah, it's also possible to score the time periods in which activities higher or lower than specific thresholds. So you can split out your data in different ranges. Yeah, and then you have, as I said, the statistics. So I think on the whole, activity parameters can be easily applied in this existing software for this application.
Yeah, I think that the result was really nice. Yeah, the result was really nice, despite the low quality of the video. I apologize for this, but as Luca said, it was done with a cell phone. The problem, the most important problem we had was the light, because we have the water surface reflecting the light. So we are thinking about how to improve the setup and the light to avoid the reflection. And so next year we will probably try to perform a new videos. And we are also investigating routes for individual identification of fish. I'm quite skeptical about putting some beads or something colored outside the fish, because sea bass are really aggressive and they will eat anything I put on the fish. But we thought about this VI tag, which has visible implant elastomers that can put subcutaneously in some clear part of the fish, and maybe we can be able to track individually the fish. Even if it's not so important, according to my experience, I prefer the group, the shoal activities, but we will investigate also this.
Thank you, Anna. We must move on because the clock is ticking. So thank you very much. This was a very short experiment, a preliminary study showing group-based tracking, so no identification. And our next speaker will show a few slides about individual tracking in rainbow trout, which is possible. Dimitri.
It's okay?
Yes, go ahead.
Yes, thank you, Lucas. Dimitri, I'm manager of fish facilities in infectiology unit at Riojosas. Today with Fabrizio, we are going to...
We are waiting for you to speak.
You heard me? With Fabrizio,
we hear you, but a bit far.
Very far in the background, yeah.
Otherwise, maybe Bertrand can speak on your behalf.
First test on result.
Okay, he's back.
Today, all of observation during experiment are done by technicians.
No, that sound level is too low.
Mobility, erratic swimming, as you can see on the picture. Use discussion. Technicians saw very marked differences between control and infected group.
No. Okay, I don't hear the sounds, so I'll summarize. We see two groups of rainbow trout here showing different behaviors between control groups and infected groups, and traditionally these behaviors are scored by human observers, but that is subjective. And we want to automate that to not only be more time saving, but increase the objectivity of the measurements. And therefore, we're using computer vision to track the fish individually, and that's being shown in these slides here. We're using colored beads
to determine
that are applied to the dorsal fin, giving each animal a unique
Lucas, now we even have difficulty
color
to hear
marker
you.
that can be seen by a video camera.
I think the videos take up too much bandwidth.
Yeah.
Okay. Well, then well, does not require any oral comments. They just speak for themselves. So we just have to watch. And then Fabrizio can step in again to give comments on this video. It's only 2 minutes.
Yeah. Here we applied individual tracking. Can you hear me?
Yes?
Yeah. We take a different approach. We are interested in what the individual is doing. And of course, if you track individuals, you can also get far more detailed data about the movement of the fish. For instance, to know how much distance, how long is the distance covered per unit of time. And so for this approach, you need to mark them. And we use here beads. In this case, it's possible because the animals are not that aggressive. So Etovision can track the corals. In this case, we tracked 5 individuals. The sixth one was not possible to follow because it has a white bead, and the white is not good for tracking with this methodology. But what you see here is the result. Each individual basically is a separate subject. So we separate the statistics of distance moved or distance from other subjects. So for instance, you could imagine to see whether there are individuals that stay further away from the group, or they are slower, they are faster, and you can compare the group itself or different individuals. Like in this case, I compare 2 individuals, or I can compare groups as a whole. So I basically merge the data of all the individuals between the different groups.
Okay, thank you, Fabrizio. Sorry for the video hiccups, but I think we got to see it quite well. Dimitri, Bertrand, thank you very much for making the videos. Fabrizio for analyzing them. We're already moving to the end of this session. We have shown you the 5 different pilot studies, and now Ben Loke of my company, our technical project manager in VetBioNet, is going to present a few lessons learned from these studies. Ben.
Yeah, thank you, Lucas.
Let's keep it fast.
Yeah, based on the pilots that we carried out so far, we compiled a number of lessons learned. It's really essential to prepare the technical system in advance, and that means it's really key to have direct communication with the partner, not only the researcher but also with the technical staff, to discuss a lot of details. Like we need the floor plan of the animal room, but also of the control room where the computer is, and pictures of the different rooms to assess the installation of the hardware and the visibility of the whole system. And as David also mentioned in his presentation, like you see here in this animal room, you have a blind spot, also caused by the rack over here where the animals cannot enter. You carefully have to select the location of the anchors. So that's crucial information. What's next is of course also to prepare the hardware itself. Enclosures should be made dependent on the requirements of the pilot itself. During the study of at the Pirbright Institute, on the last moment we created, or the Pirbright actually created enclosures to have the anchors IP65. And also the small tags have small screw holes which should be sealed to have it decontaminated easily afterwards. Next, please, Lucas. If we look to more specifically planning the hardware itself, it's essential to assess what is possible, like mounting the anchors to the walls. Is it allowed to screw in the wall, or do we need a rubber plate which is glued to the wall instead of these screws, so that the anchor can be placed on this rubber pad, for instance. Also cabling and all the details about when do you want to do this is of course essential to plan in advance. Also the precise equipment which goes in the BSL 2 or 3 containment envelope must be distinguished separate from the equipment what stays outside the containment, and not only the hardware which is needed for tracking in the animal room itself, but also the tools that you need in this room to calibrate, for instance, the system must be listed in advance and also prepared in advance to have smoothly process. What we also learned is that remote access is needed. Ethernet access to have remote support from the Noldus staff is of course also essential. Yes, next. The installation of the whole equipment, especially for complex, more complex systems like the TrackLab ultra-wideband system, requires quite some time. We can save a lot of time if a lot of cabling and mounting these anchors can be done in advance. Calibration is very a precise measure to perform, and can only be performed by the Noldus staff, so that should be taken into account as well. And also specific procedures like validation of the calibrated area requires certain patterns to walk by the staff in the room itself, which is often very difficult to make clear what exactly is needed, what kinds of patterns must be walked to have it validated. Other systems, although, can be more simple to install, and with remote support we can go quite far. Yes, please, next. If we look to the impact of COVID-19, then we saw that for the simple systems like camera observation systems or video tracking, that can still continue with remote support if needed. But more complex systems, like I mentioned, the TrackLab systems, we cannot perform this due to the governmental and organizational rules. As we know, it's simply not possible during the lockdown period and are postponed or even cancelled. If we look to the pilots we carried out and we had planned for past year, we had to postpone. Then in Morden, for the Morden Institute, India, EV, and INRA New Zealand was planned for January. We all had to postpone. So that was a serious impact. That was the last slide.
Well, thank you, Ben. I've asked the two remaining speakers to shorten their talk a bit so that we are wrapping up. I apologize for going over time. We apparently were a bit off with the calculations. So Alassane Keita from Ansys and Ploufragan is going to give us a brief update on his work with telemetric monitoring. We are switching now from behavior to physiology, body temperature. Alassane, floor is yours.
Yes, thank you, Lucas. Do you hear me?
Yes, I hear you.
Yeah. Okay. My topic is about evaluation of a telemetric sensor, which is called ANIPIL, for continuous monitoring of body temperature in chickens. And we did two experiments. First, for the safety of the system, and we also use the system during an infectiology experiment. Next, please. Okay, in terms of presentation of the tested device, it is very small. You can see the characteristic of the device. It is composed of an activator for identification of each capsule. For example, for sampling the frequency, it is possible to use, for example, every 30 seconds, every 2 minutes, or 5 minutes, or 15 minutes. It is not a real time monitoring, since we need to use a monitor for data recovery. So each capsule stores the 2000 last data. The capsule use wireless technology to transmit data up to 3 meters to a monitor. Next, please. Okay, for the first experiment, we use 20 SPF chickens, ANIPIL animals, and also control animals in two pens. The study lasts 14 days, and from day 0 to day 14, we took a cloacal temperature on all animals once per day using medical device, in addition to the telemetric system for the ANIPIL animals. Next, please. For the second experiment, we use a very virulent infectious bursal disease virus and we have two groups, mock infected animals, and the second group is infected. And we use also clinical scoring for IBDV. Next, please. In terms of presentation of results for safety experiment, we have no macroscopic lesion in the gizzard for any animals. So I forgot to precise that in terms of administration of the capsule of animals, we introduce the capsule into the esophagus, and a soft massaging ensure movement of the capsule down the crop to move in the gizzard, and the capsule remain in the gizzard. So the safety was also demonstrate because we have no statistically significant difference between the mean weights of the two groups over time. Next, please. Yes, and for the challenge experiment, you can see temperature variation likely due to the circadian rhythm, and in bold, it is you can also see a dramatic hyperthermia following infection for infected animals. Next, please. I think it is last one. So, in summary, in terms of testing, the safety of the anipil system was demonstrated because oral administration was easily performed. Chicken that were given the capsule had a similar graph compared with control chicken. And interestingly, for our viral challenge with a very virulent IBD virus, we highlight the interest of the system because it was more sensitive and more objective and gives also more objective information when compared to clinical scoring only. I think it's that's all. Thank you.
Thank you very much, Alassane. This was the first telemetry study that we did on physiology measurement in work package 9. But there's much more to choose from because there are so many technologies on the market and so many different animal species. And that's why we started a telemetry survey. And that's the last some presentation of today's session given by my colleague Leslie Vedder, our assistant project manager at VetBioNet. Leslie, the floor is yours. I don't hear you. Are you speaking? Then you have to turn on your microphone.
Do you hear me now?
Yes.
Ah, okay, good. Perfect. Had to set another microphone. Yeah, so I will summarize quickly all the vendors that we approached to help us out with solving the telemetry in body temperature. Because at Noldus IT we only have systems for behavioral monitoring, but not for measuring body temperature. And we want to solve this by finding another party that helps provide this technology. We approached 12 manufacturers. You see that in the table. And from those 12, 8 replied. And those were also really happy to help us out in this project. And what we wanted from the vendors was that they have a solution that in real time measures body temperature that is wireless. So that the receiver can be set inside the pen or outside the animal pen, and that retrieves it in real time. Yeah, so that was asked specifically, and we also showed use cases per animal group if they have a solution for those animals, because we have such a variety of animals. We got quite a lot of feedback back from that. So the use cases that we provided for the vendors was cattle, pig, sheep, ferret, hamster, and chicken. And in the next slide, you will see the general information that we asked each technology company. And that that was also that the technology will work in group housing, and that can be used in together with other software programs. And the next slide, you will see an example of the use case for cattle. And you see that we also ask other species. Next slide, please. Yes, and here I summarized the results. So 8 companies want to continue in this project. And we have different solutions for different animals. So the top four are already companies that work with animals, and the bottom four solutions are human solutions. But they indicated that it can be used for animals, and for HQ Inc., there's a question mark for ferret because they haven't experienced in ferrets yet. Yes, next slide, please. In the next 3 slides, I will show some examples of how the systems will look like for different animal sizes. So for a large animal size, we have a Vitalo Plus solution. They can monitor the animals from quite some distance, using different anchors also for reception. And the key features from those systems is that you can program the interval via the computer. So you can set it on, you can enable it, disable it, and set it 2 times. It's in 1 week you want to sample every 10 minutes, or the next week you only want to set 1 hour. That's possible, and you can see also the battery life. And the next slide, you will see an example from medium-sized animals inside pens. So you will see that you need more receivers also when you use different sizes of pens for different systems. And also here, you can program the interval and enable, disable the sensors. And the next slide, you will see solutions for smaller animals. Because of the smaller size animals, the weight and the size of the product also needs to be smaller, and this makes more difficulties with the reception of the solutions. And the next slide, you will see a summary of the products. And so you see that there are a lot of different products, also different ways to apply the solutions. And also some of them have activity sensors inside the products. And also the receiver differs quite a lot in all the solutions. And this is what I explained due to the weight and the size of the solutions. I also mentioned what you see is that the maximum distance differs between, for example, with Data Science International between 3 and 5. And the vendor indicated that this is due to the size of the animal and if there's other objects inside the pen. Next slide, please. So this is what we all collected in the last months. And in the coming period, we will organize a vendor meeting with all the discussion groups again. We present all the results, what we collected so far, including the pricing. Then we will send out a survey so that everyone can indicate if they have experience and which kind of solution they want to try out. And then we will organize a vendor meeting where all the vendors can present their technology to all the partners. And afterwards, we will make a decision to where the technology goes to and in which studies they will be used. And afterwards, we will test the technology and share the results with all the vendors. So this was my short summary of what we did and what's coming.
Thank you very much, Leslie. You've only been with our company for 1 month and you already did a lot. Congratulations. So my final slide is for me, and then really we have to stop this session. The wrapping up slide. So there's a lot going on in Work Package 9.2. I'm very happy with the active engagement of the discussion groups, the 60 people convening on a regular basis, lively discussions, a lot of inspiration. I get a lot of positive feedback from my own colleagues, from the scientific partners. So happy with the way we're moving forward. With respect to behavioral monitoring, I think we're moving towards having solutions for every species, different technologies, computer vision, auto wideband tracking, auto tracking, which will eventually work. I'm confident. For the physiology, the telemetry survey is almost done. and we have a lot to choose from. And there again, happy with the positive interaction with the leading manufacturers. They're really constructive and happy to support us. But of course, COVID-19 was a real setback. Many of these pilot studies delayed, so we will need some more time to reach all the goals in our ambitious list for the VetBioNet project. But that's a common problem for all the work packages, I think. Sasha, really sorry for running over time so much. I hope it was worth it, and I quickly give the microphone back to you.
Okay, thank you, Lucas. I'm sorry that we have to skip the questions, but as it's stated in the chat, you can ask your questions in the chat. I think it's clearly visible to everybody that this is quite a success story. So thanks a lot, also Lucas, for being so active. I think everyone appreciates all partners who are involved. That's really great work. So Tobias, it's up to you now. I'm sorry also for the delay, but I see that most people are still connected, and I hope they will stay with us.
Okay, so, oops. I share my screen. But I think it's not in the presentation mode, right?
No, it's not.
Yeah,
now it is.
Now it is. Okay, cool. I will present the work on creation of novel animal cell lines. The presentation will be divided in 3 parts. I will give a short introduction about the technology that we developed at InSCREENeX. And then I will hand over to Paul von Wageningen, so from the Institute Wageningen. He will present work that he did with cells that we immortalized, and then afterwards to Ignacio from InRAE, who will also present work with cell lines that we developed here in InSCREENeX. So, oops. Okay. The reason why we are doing the immortalization is basically shown here. This is data from Cellosaurus, so only listing the available cell lines for different species, and altogether there are only 1,700 cell lines from all these species here, and this in comparison to humans and mice, you see it's a dramatic, significant difference. And this is something we would like to change, because we know that we need for all the different animals also nice in vitro tools to do molecular studies, and so on and so forth. So, and the thing is that, of course, from animals you can relatively easy isolate primary cells, but the problem with primary cells is that they are not available, or you can only generate a low number of primary cells, and therefore for lots of research purposes you don't have enough of them, and therefore we developed the InSCREEN approach, and this is based on a gene library which is comprised of 33 different genes, and these 33 different genes are integrated into 33 different lentiviral vectors, and these 33 different genes, they target different signaling pathways like cell cycle, apoptosis, stemness, etc., etc. And what we do is we usually, not usually, we use this library. In fact, primary cells from any organ, from any species, and then simply do nothing else than expanding the cells. And what happens is, as this transduction is a random process, not all of the transduced cells will become immortalized, but some will simply die off because they got integrated gene combination which is not suitable. Others are not infected, and of course we are simply by expanding the cells, screening for those primary cells that become immortalized and can be expanded unlimited. Okay. And this worked indeed for a lot of different cell types, ranging from lung epithelial cells to hepatocytes, osteoblast, endothelial cells, osteoblast, chondrocytes, as is here, intestinal cells, and so on and so forth. And shown on the right, you see depicted some of the cells that we already established, as the cell types that we established from humans. So this technology is more or less universally applicable. And but my presentation is somehow stuck.
It's still moving on my screen.
Okay, that's good.
The cursor is moving.
So then you take over and sure.
I talked enough.
Okay. No, it won't. Okay, so I just give you a few examples. So because what I just showed you is that we can immortalize these cells and that they are unlimited expandable, but this is not the aim. The aim is to create really cell lines that show functionality. And here are bronchial epithelial cells that were cultivated under air liquid conditions, which fully differentiated into a nice airway epithelium. And also quite nice is when we immortalize smooth muscle cells, they show functional calcium signaling. And when we cultivated them on a 3D cell, on a 3D matrix, you see that if you stimulate this 3D matrix with the cells, the matrix contracts. So this is something, of course, very specific for smooth muscle cells. And another example is shown here for astrocytes. Again, we have a nice calcium signaling when stimulated with baclofen, and of course with image tracking you can nicely generate data for single cell analysis. And we have within this library, as I mentioned, 33 different genes, and these are mostly human. Some of them are also from murine origin, but we then did a search against a homology search and found that most of the genes are pretty conserved throughout the different species, and therefore we were pretty sure that we are able to transfer this technology also to other species. And indeed, it worked out. So we have now really a nice zoo established. So not of immortalized animals, but immortalized cell lines from all these animals. Unfortunately for llama and muskrat, I don't have a picture, but so we have now from all these animals developed cell lines. But you see that most of the, not most, all of those are of mammalian origin. And so we ask, is it possible to also generate cell lines from avian species? And we used their chicken. And another thing that is still where we don't have any success stories is we don't have any cell lines generated from the blood system, and no matter which species. So, and these were two things that we try to develop through VetBioNet. Starting off with the chicken cells, and this is a pretty short story because we tried 3 different cell types which are mentioned here, and in addition 2 other cell types which I'm not allowed to disclose here, but altogether 5 different cell types from various stages, so from embryonal chicken to adult chicken. And what we got is no immortalization, at least not due to our InSCREEN immortalization approach. And so for the embryonal fibroblasts, we got immortalized cells, but these were spontaneously immortalized, so not due to our nice technology. So we've checked whether we can transduce the chicken cells with a simple GFP vector, and with our original protocol for infecting these cells, we got a very low efficiency of transduction, but also after a modification of the transduction protocol, where we got a really nice infection of nearly every primary chicken cell, we got no immortalization. So to make a long story short, we can transduce the chicken cells, but up to now we don't know why we haven't been able to immortalize these chicken cells with our technology. So it seems at the moment that the mammalian space is okay for us, but anything outside the mammalian space is at the moment not doable. So we had, I think, from Roslin, I'm not sure, they provided us monocytes from sheep. And we tried several times with our old transduction protocol to transduce these cells with a reporter virus. And we saw no transduction at all. And of course, due to no transduction, we also see no immortalization. Therefore, we went for a new transduction protocol, which we developed over time. So this is from here to here. This looks pretty easy, but it took us nearly, I don't know, two years. So we have now a really nice transduction efficiency. We don't know yet whether the cells are immortalized, so this is still ongoing, but at least we have hope here that we now can see that we can transduce the cells. And we are pretty sure that with this first step into the cells, we will be able soon to immortalize also cells from the blood system, which would be a huge success story, at least for us. And with this, I want to stop my presentation and hand over to people who have done nice things. So I presented more or less the things that were not so successful. So I leave this more successful stuff to Paul and Ignacio. So the first presentation is from Paul, and I would hand over to you, and please indicate when I should switch the slides.
Yes, I hope you can hear me all.
Perfect.
Okay. Yes, thanks. I will give some overview about the primary cells we are working on, and we focus on ovine primary cells, so sheep cells. And in this project, so to say, we also want to bridge virology and bacteriology. Myself, I'm mostly interested in primary ovine cell cultures for assessing virus susceptibility for Bunya virus and flaviviruses, for arboviruses in this, so to say. And we are mainly interested in the cells which are in vivo also mostly susceptible for these types of viruses, and these are liver cells and cells within the maternal-fetal interface. And to bridge to the bacteriology, there are also a lot of bacterial pathogens which are able to replicate at, for instance, the site of the fetal-maternal interface. And having additional tools is really important, because there are not a lot of sheep cell lines available so far. And this will also facilitate assessing host responses in these cells, so without the need to always move to the in vivo situation. Next slide. So how did we start this project? So we obtained primary cells from a pregnant animal which was euthanized. This animal contained one male fetus, and from this from the liver of this fetus, we cultured primary cells, which we were then sent out to InSCREENeX, and we also cultured the placental cells. And very briefly, I would introduce you to all the cell types within the ovine placenta. So you have fetal trophoblasts. So this is these are the epithelial cells from the fetus, and lining to these fetal trophoblasts, there is maternal epithelium, and just between these epithelial cell lines, this is the bridge between the mother and the fetus. And there are a lot of other cell types present there. And interesting, this is also, these cells are what we showed in vivo experiment. These cells are highly susceptible for arboviruses, probably because also the innate immune responses there are different, are lower to say, or different compared to other cells in the body, because the fetus is not should also be protected from the immune system of the mother. So next slide. So in the end, we successfully received 7 liver cell clones back from InSCREENeX, and 15 placenta cell lines. And these cell lines we used, of these cell clones, we used for further characterization. Next slide. And some of this characterization involved assessing susceptibility of these cells for a larger panel of arboviruses. So we work on Univirus, Smallberg virus. Those are viruses which really efficiently cross the transplacental epithelial barrier. Middleburg virus, Wesselbrand virus, Alpha virus, and Flavivirus, and Rift Valley fever virus. And there was some heterogeneity within all the cell clones, as expected, and some cell clones really showed that they were highly susceptible for this panel of viruses, and this, of course, these cell clones are of most interest. Subsequently, we next slide. Subsequently, we also assessed really virus growth in time. So what you just saw was staining of the virus at one moment in time, and we successfully showed that a wild-type virus, so a wild-type univirus, is able to grow efficiently on the ovine hepatocyte cell line 3. However, a mutant virus which lacks the interferon antagonist. This is it's a delta NSS protein. This protein is able to antagonize these interferon responses. Is not able to grow efficiently in these cell lines compared to conventional Vero cells, in which we do not see major differences between these virus variants. So this gives us a better readout for assessing potential virulence factors. So next slide. At this moment, we are still focusing also on the further cell characterization, which is of course very important. So we have a lot of different clones, and some might be really the clones we are mostly interested in. So the epithelial cells in the placenta. But we have to still confirm for some of the clones whether they are finite or continuous, what kind of differentiation state they have, and whether they are really stable over time genetically. So next slide. For some of these clones, we already obtained very good results to our opinion. For instance, the ovine placenta cell line 16 shows cytokeratin expression upon differentiation, and this is really an important feature to what you could observe in maternal epithelial cells. So we hope with further characterization that this clone turns out to be turns out to be originated from the maternal epithelium. And interestingly, we have one liver clone which shows characteristics of liver sinusoidal endothelial cells, besides just normal hepatocyte, which we also think we have. And these cells are really highly specialized cells in the liver, and they play major roles in immune responses in the liver. So on the next slide, I will show that what our strategy is considering the further characterization of these cell lines. We plan to do RNA-seq on the RNA isolated from these cell lines and compare all the expressed RNAs with the gene panel which is available, and see whether we could really pinpoint and see that some characteristic genes are expressed in these cells. Hopefully, I think within 3 or 4 weeks, the results will be available, and I will be able to say something more about it. And in addition, we also aim in the next year to develop a more complex cell system by combining maternal epithelial cells and the fetal trophoblast in a transwell system. So in this sense, we can more or less mimic the barrier also as is present in vivo, and hopefully we can use this model to reduce animal experimentation and to assess much more variables as you can do with the use of animals. Next slide. This was now I have to hand over to Ignacio.
Oh, no.
Hello?
I
Can you
have
listen to me? It's okay? The sound?
yes, I have only to switch again back the presentation. So floor is yours.
Okay, thank you very much. I'm going to talk about the work we're doing with Tobias and InSCREENeX. In my team, myself and my team, we're interested in the bronchial epithelium in the airways. Especially me, I work in the pig. So this is a very heterogeneous epithelium with secretory cells, mucociliary cells, etc. Well, this is developing this tool is really important, especially for as evaluation the host immune responses at the mechanistic level. Also, is very important what they said before. For animal welfare, it really makes the life easier and it's better for animals if we can have a tool that is it can be replicated and we can work with them without the need to go and kill animals and produce new primary cells. Next slide, please. So what we did, we first produced in air liquid interface an airway epithelium. What we have is in the photo, you see this is the lung that we dissect, and we take the bronchial epithelial cells and we put it in an air liquid interface. Then we check that they develop an epithelium. We can see mucin and tubulin expression. And we just pretty much we send that to Tobias and he give us back 7 different clones. Next slide, please. From the 7 different clones, we did a quick screening to see which one had a good immune response to some toll-like receptors, and I decided to start working with the T3 line. So we saw first we characterize a little bit the growth kinetics, they double almost every day, more or less. Then we measure the transepithelial resistance. The transepithelial resistance in the right is increased when we put in a liquid interface and we differentiate the cell line. This means that they're producing tight junctions. Next slide. Then with this an immunohistochemistry, we're testing the cells non-differentiated and differentiated. So when it says non-differentiated, that they are not in the air liquid interface, to see what is the process, what happens during the process of differentiation of these cells, and then the differentiated. You see on the left side, you have the non-differentiated stained with mucin and tubulin, but we don't see any mucin. And then when after 2 weeks of differentiation in air liquid interface, it seems that the cells are able to, the secretory cells differentiate and at least are able to produce mucin. This is MUC5AC. Next slide, please. Then we did some quick qPCR for some of these markers, and you can see then on non-air liquid interface and the air liquid interface in, for example, there is a big increase in the expression of MUC5AC and TLR4. The cells are able to differentiate and they change a little bit during differentiation. Next slide, please. Also, we're working with influenza, pig influenza and avian influenza, but in pig, in our team, the pig is really important for the influenza as a mixing vessel. In the primary cells, the pig is able to express 2 types of sialic acid receptors, the alpha-2/3 and the alpha-2/6. That means that the pig is able to be infected by avian and swine influenza. So we wanted to check if these cells express the 2 types of receptors with some lectins, with the lectin MAA that recognizes the stain, the alpha-2/3 sialic acid, and the SNA stains the alpha-2/6. And we can see in our cells that we have alpha-2/3 sialic acid on the left, the MAA, and there is this is for the avian influenza receptor, and the SNA. There are much more. So these cells, after differentiation, they replicate very well what it will be the primary cell, cell line in pigs for these lectins. Next slide, please. And then this is a work in progress. This is just some preliminary data. We are doing much more experiments about how is the immune response. Actually, I have made a lot of data from primary cells, and it seems they are able to respond very well to, for example, this is with flagellin for toll-like receptor 5 expression. This is very similar to the data I'm observing in primary cells. This is something that I was using another cell line before, I was having problems because they didn't respond as I was used to observe in primary cell. But with this cell line developed by Tobias, they are pretty good for my sense. Next slide, please. Also evaluated the response to toll-like receptor 5. How does it work, the signaling pathway? What happens when you stimulate toll-like receptor 5? NF-kappa is activated and it goes into the nucleus, and that will start transcription. After a while, there's an oscillation, it goes to the nucleus, and then after a while it goes out of the nucleus. So what we did here is an immunohistochemistry of for P65, a subunit of NF-kappa B, and you can see the dynamics. The timing you see at the 0 minutes, this is P65 is all in the cytoplasm, so you see the dark nucleus, there is nothing in the nucleus. At the 20 minutes, you can already see a lot of the P65. When I stimulate with flyolin, it goes into the nucleus, and there is an oscillate at the 50 minutes, it's already going out, and to 120 minutes is out. So it has an oscillation similar to what is described for primary cells for a normal response to a toll-like receptor. And next slide, please. Well, and as I said, this is a work in progress. We are working now on doing influenza infection and studying in more detail the host response to different PAMs. And also we want to, I'm very interested to see if we can do some kind of single cell RNA-seq to see if we find if it is from the clone, from the cell, the T3 cell line, and how many kind of different subtypes can be differentiated if we are able to reproduce the whole epithelium similar to the primary cells. And this is all for me. Thank you very much.
Yeah, and I want to thank all the speakers, so Paul and Ignacio. And of course, it was not me, only me who did the work. So Teresa and Christina did it for the cell lines that we developed here, and for Wageningen, you see all the people involved, and also for INRA. You see on the right side all people involved in the study, and yeah, that's it for the presentation. Now I would like to go back.
Okay. I've seen there are some questions in the chat. I don't know if you have followed it, Tobias. If not, I can read it. Ryan has a question. I will simply read it aloud. Or Ryan, do you want to ask your question directly? Perhaps it's
sorry, I actually asked that before that last presentation, and you kind of answered the question. But I guess sort of a supplementary one. Which species have been looked at with regard to using this modeling respiratory tissues?
You mean the last study was in pigs?
And is it only pig that you're looking at, or are you looking at trying to apply this to other species?
I think we have also done immortalization of bovine respiratory cells, but yeah, I don't know.
I don't know if Fabienne is perhaps connected, because she was involved there in the immortalization of bovine bronchial epithelial cells. Perhaps she can comment on this.
Yes. Hi, hello.
Hi, Fabienne.
Yes.
Hi.
Yes, so we furnished to Tobias bovine primary lung cells. It was more alveolar cell than bronchial cells.
Okay.
So we are currently screening the different clones that Tobias furnished. So, and we are trying to characterize them to see if we have more pneumocytes or which type of epithelial cells we got in the different clones. So it's ongoing work. And of course, the aim afterwards is to come with virus or bacteria that target the bovine lungs.
Okay, thank you.
That's all going.
And then there's a question by Martje. I think it's a specific question for you, Tobias. What types of gain, loss of function does immortalization by SciScreen bring apart from eternal multiplication?
Oh, yeah. I'm afraid I'm not fully understanding the question.
Okay, maybe I can try to explain. One would expect certain properties of cells to be selected for in the selection process. And people, of course, want certain characteristics of the primary cells to be retained through this selection process. Can you tell anything more about it? Are there typical features connected to that selection strategy?
No, I don't think so. And actually, we try to get rid of any bias. So we have now started to really create clonal cell lines with the last immortalizations. So before we had this, and I think Paul also mentioned it, these cell lines are very often polyclonal. And the good thing with the, for example, with the approach that we did with Paul and also with Ignacio is that we received a mixture of primary cells. So it was not a pure population, but it was basically comprised of several different cell types. And we throw the cocktail on this mixture. And then it's only up to us to identify those cells or those cell types that we are interested in. And up to now, I would say it's really a matter of the selection scheme. So as long as you have a good readout for the cell type you're interested in, you can generate the cell lines. So I don't think there is a common feature for all the cell lines that we developed so far.
Okay, thank you. So you have to define the type of functional cells that you would like to convert into a line.
It would be helpful, yes. So this is something that we definitely see. So the more the end user of the cell line knows about the cell model he wants to develop, the easier it is for us to really develop the cell line for the customer or for the researcher.
That's really great.
Maybe to add, this is Paul. I think it is also very difficult to get a homogeneous primary cell line of primary cells, which you could use for immortalization. So if you have low passage material, you will have always a lot of different cell types in this. And if you start with this low passage material, you will end up with purified clones in the end. And then the trick of the most important part will be the characterization there. So can you really characterize as that good that you can state, okay, this clone was this type of cell or another type of cells? But there is still I also believe that there is still a lot of things to learn there, because especially for ruminants or other farm animals, the tools are not, the toolbox is not so large. But we hope with the more systemic RNA sequence technology to be able to characterize them better, but that's to be awaited.
And
I
this
think
is something
Lonneke might have a question because she connected her camera earlier.
Yes, and I really, really enjoyed your talk. Thank you very much. It's a really nice system. I was also very pleased to hear that the chickens didn't work. That's very good to hear.
for us,
We've been trying for a long time. What I wondered, I think the whole idea of having not a monoclonal cell line, but a polyclonal cell line with multiple different cell types, more similar to an organoid, is a very attractive idea. Have you followed these polyclonal cell lines over time to see whether certain cells stay proliferating while the others die out? How stable is a polyclonal cell line?
This is, I think, the big disadvantage of the polyclonal cell lines, that you have this Darwinian mechanism, so this survival of the fittest, the cell clone that is proliferating the strongest will overgrow the others, and therefore it is really hard to keep a polyclonal cell line polyclonal in a certain mixture. So you mentioned organoids. So we had, and also with these bronchial epithelial cells, it looks like we immortalize the adult stem cells, so the basal cells for the bronchial cells. And if you then stimulate the cells in a correct manner, then they differentiate in the different cell types, so that you don't need to preserve the mixture of the heterogeneity of the bronchus. And the same is true for the organoids from the intestine. So we immortalized, some kind of stem cells. When put back into matrigel, they differentiate into these fancy spheroids, and then later on to these, I don't know how they are called, but you know these. I hope it was not too wordy, my explanation, and I don't know what else to say.
No, thank you very much. It's very clear.
Can I have a comment here? I have my camera. Okay, you can see me. For example, what we saw in the cells that you provide to me, it seems that it may be possible that you have immortalized some kind of basal cells or something, because it is clear that after differentiation, a timing differentiation, the cell type, the epithelium changes. We start to proliferate the mucin. We
can hear you, Ignacio.
Tobias is there?
I can hear you well, Nacho.
Yeah. So yeah, I think also that the polyclonal epithelium is
yeah, I think my connection is now a bit slower. Therefore, I switch off the camera.
So yeah, I agree that is a very good. I really appreciate the polyclonal, the possibility. If we want to produce an epithelium, a real bronchial epithelium, is not like a kind of single cell. Well, ideally, we produce the basal cells immortalized, and then from there we can differentiate as the idea. But I don't know.
Okay, is Tobias still connected? I think yes, he might have some issues, but anyway, I think we must now terminate the session. Would like to thank again Tobias, Ignacio, and Paul for the presentations.
Okay.
Okay.